In the last session, we have discussed about the development of brain and three basic concepts. The very first one is development of your brain vesicles, followed by the formation of flexures and the development of ventricular system. After that, what was the topic left? It was histogenesis of your neural tube. Okay. So, what are all the types of brain vesicles we have seen? In which week we can see the development of brain vesicles? 5th week, okay, starting from the 4th week to 5th week. So, what are all the brain vesicles we have seen? First, we have seen the development of primary brain vesicles. What are all the primary brain vesicles? Okay. So, in the initial stage during the development of brain vesicles, we have seen the development of three brain vesicles. Those are prosencephalon, mesencephalon and rhombencephalon. Okay. So, these three primary brain vesicles will give rise to your secondary brain vesicles. Once the secondary brain vesicles formation takes place, we can totally see the how many brain vesicles? 5 brain vesicles. What are those? Okay. So, this is telencephalon, diencephalon, mesencephalon, followed by metencephalon, myelencephalon and spinal cord. Okay. Your telencephalon will give rise to cerebral hemisphere and your diencephalon will give rise to thalamus, hypothalamus and epithalamus. Mesencephalon will give rise to midbrain and the metencephalon will give rise to pons and cerebellum. The last part, that is myelencephalon, will give rise to medulla oblongata stage. Okay. And what about the flexures? What do you mean by flexures? Flexures are nothing but the folding of your developing brain part. Okay. So, how many types of flexures we have studied? We have studied 4 types of flexure. The one that takes place between your spinal cord and the hindbrain part is called as cervical flexure. Okay. And the one that takes place between your metencephalon and myelencephalon is called as pontine flexure. Okay. The one that takes place in the middle of your mesencephalon is called as mesencephalic flexure, right? And the one that takes place between your telencephalon and diencephalon is called telencephalic flexures. Okay. What about the ventricular system? What do you mean by ventricles? Yes, space. Anything else? So, basically your brain and spinal cord are developing from the neural tube. Okay. So, your neural tube is a hollow structure. It have a space. Later in the brain and your spinal cord, it is going to develop as a ventricles and central canal. So, what are all the ventricles we see here? 4th ventricle where? First we have lateral ventricle, then followed by 3rd ventricle, then we have aqueduct, that is in the mesencephalon region here, cerebral aqueduct. What is the another name for cerebral aqueduct? What is the another name for the cerebral aqueduct? Secondary? No. It is called as aqueduct of Selvis. Okay. And the last one is 3rd ventricle, followed by Fourth ventricle, okay. Followed by central canal in your spinal cord region. Okay. So what fluid runs in this ventricle? It is the CSF. CSF is the fluid that runs in the ventricle. So where we can see the formation of CSF? What are all the components of CSF? Your CSF is made up of what? Meninges and CSF is finished, right? Lecture class. Meninges is finished itself. CSF is also finished. Your previous batch students said like that. So tell me what exactly is CSF? What are all the components of CSF and where we can see the formation of CSF? What is the function of CSF at least? Okay. Friction, okay. Act as a lubricant, okay. And it helps to avoid the friction. Say it louder. Okay, everyone just go and check the components of CSF once. It is directly given in your textbook or you can see your previous lecture, you will get to know. Okay. Your CSF is mainly produced in your lateral ventricle. So whatever the CSF produce here in your lateral ventricle region should run until your central canal part and also to the surrounding area. So what is the surrounding area we can see here? What is the surrounding area we see here? What space is present? Arachnoid space, okay. Pia mater, okay. Dura mater, okay, fine. Subarachnoid space, fine. Okay. So whatever the CSF produced in the lateral ventricle will run until your central canal through the foramens. Okay. Foramens are the gateway between these ventricles. So what are all the foramens we can see? The foramen present between your lateral ventricle and third ventricle is called as interventricular foramen. Interventricular foramen. Okay, which is also known as foramen of Monro. Monro, M-O-N-R-O, foramen of Monro. Your third ventricle communicates with your fourth ventricle through the cerebral aqueduct. Okay. The last one is fourth ventricle, it should communicate with your central canal. Remember the point, your fourth ventricle is basically continuous with your central canal like this. Okay. So there will be no foramen. Instead of foramen, on both the side between your hindbrain and the spinal cord, a very small dilation is present. This dilation is called as terminal ventricle. Okay. So this is all about the foramen of your ventricular system. Your lateral ventricle communicates with the third ventricle through the foramen of Monro, and the third ventricle communicates with the fourth ventricle through the cerebral aqueduct. Your fourth ventricle is continuous with the central canal, so there will be no foramen. Instead of that, there will be a small dilation which is called as terminal ventricle. Okay. Any doubts in this? Any doubts? Any doubts? So this finishes your introduction classes for the development of brain. We have discussed about the brain vesicles, flexures and also ventricular foramen along ventricular system with the foramens. Any doubts? Okay. Next we will discuss about the clinical conditions. Remember this diagram? What is this? This is your amniotic cavity and yolk sac. This is your neural plate. Okay, and later this neural plate is going to develop as the neural tube part. Okay, like this. Whatever the neural tube produced in the initial stage will be in contact with your amniotic cavity. What is the reason? Increase in the your neural tube developed in the initial stage will be in contact with the or it is communicating with your amniotic cavity. What is the reason? In which week we can see the development of neural tube? We can see the development of neural tube in the 3rd and 4th week. Okay. So during this week your blood vessels are not fully developed. For that reason your neural tube is dependent on your amniotic cavity for the required nutrients and nourishment. Okay. So this point, this is called as anterior what? It is called as anterior neuropore, and this point is called as posterior neuropore. Okay. And your anterior neuropore is present at which somoid level? These points are already discussed. At which somoid stage we can see the anterior neuropore? 4th. In the 20 somoid stage, we can see the anterior neuropore, and 25th somoid stage, we can see the posterior neuropore. So total how many pairs of somoids are there? How many pairs of somoids are there? Total there are 44 pairs of somoids are there in the developing embryo. Okay. So by the middle of 4th week, your anterior neuropore will get separate from your amniotic cavity. Okay. And by the end of 4th week, your posterior neuropore will get separate from the amniotic cavity. This is the normal development process. Okay. In case your anterior neuropore and posterior neuropore fails to get separate from your amniotic cavity, it will lead to certain clinical conditions. What are those? If your anterior neuropore fails to get separate, it is called as anencephaly. Okay. And if your posterior neuropore fails to get separate, it is called as rachischisis. Rachischisis. Rachischisis is one of the severe form of spina bifida. What is spina bifida? What is spina bifida? Say it louder. Okay, right. Okay, these are the two important clinical conditions you should remember for the defects of neural tube. Okay, the first one is anencephaly and the second one is rachischisis. It is the severe form of your spina bifida. Okay, we have one more important condition called hydrocephalus. Hydrocephalus. So, what do you mean by hydrocephalus? Accumulation of fluid. So, in this particular case we can see the excess accumulation of CSF in the ventricle region or in the foramen part. Okay. This will lead to a condition called hydrocephalus. These are the three important clinical conditions you should mention for the defects of neural tube. Any doubts in this? Any doubts in the foramen or in the clinical conditions? Any doubts? Accumulation of CSF in your ventricles and also in the foramen. Excess accumulation leads to a condition called hydrocephalus. Okay. Any doubts? No doubts? This is the notes for the foramens. The two lateral ventricles communicate with the third ventricle via interventricular foramen, which is also known as foramen of Monro. The third ventricle communicates with the fourth ventricle through the cerebral aqueduct, which is also known as aqueduct of Sylvius. The fourth ventricle is continuous below with the central canal of the spinal cord, which presents a small dilation at the inferior end. It is called as terminal ventricle. Okay. The cerebrospinal fluid is mainly formed in your lateral ventricle and it leaves the ventricular system through the foramen of Magendie and foramen of Luschka into the subarachnoid space around the brain and spinal cord. So, along with these three foramens, we have other two important foramens. They are called as foramen of Magendie and foramen of Luschka. Okay. So, your CSF will enter to the subarachnoid space through these two foramens. Note it down. Foramen of Magendie and foramen of Luschka. So, this is the picture to show your ventricles and also to the foramens. Okay. Next, clinical correlations. The first condition is called as anencephaly, failure of the cephalic part, that is nothing but the anterior neuropore of the neural tube to close, and associated defective development of the vault of the skull produces a congenital anomaly called anencephaly. The characteristic features of this condition is vault of skull is absent. So, which one is the vault of skull? Can you all point the vault of skull in your body? Okay. This region, this is called as vault of skull, not this. This or this area, this whole area is called as the vault of skull. Okay. Brain is presented as a mass of degenerated tissue directly exposing to the surface area. Appearance of a child is characteristic with prominent eyes bulging forward, and also your chin part is continuous with the chest due to the absence of neck region in this particular condition. Okay. Can you see the difference in both the picture? Stop talking. What is the difference between two pictures? Vault of skull is absent or underdeveloped. As a result, the brain is exposing into the surface area as just a mass of tissue or cell. Okay. And here you can see some difference in the neck region also. Okay. So, this is how the baby suffering from anencephaly will appear. Okay, finished. The next condition is called rachischisis, which is the severe form of spina bifida. An incomplete closure of caudal neuropore, that is nothing but your posterior neuropore, and defective development of the associated vertebral arches causes a congenital anomaly called the rachischisis. The characteristic features of rachischisis are usually localized in the lumbosacral region. Neural tissue is widely exposed to the surface area. Occasionally the neural tissue shows considerable overgrowth. Usually the excess tissue becomes necrotic shortly before or after the birth. So, what do you mean by necrosis or necrotic? Necrosis, death of the cells or tissues. Okay. So, this is how the baby suffering from the rachischisis will appear, and this is the neural tissue directly exposing to the surface area present at the lumbosacral region. Okay. So, right after the birth, these tissue cells are going to, what happened to those tissue cells? Death of the cells will take place. After that, they can, whatever the mass you are seeing, no, that can be removed by conducting a surgery. So, this is how the baby with rachischisis will appear. Okay. Finish. The next one is hydrocephalus. It is a clinical condition characterized by the dilation of ventricles due to the excess accumulation of CSF within them. It occurs either due to the overproduction of CSF or obstruction to its circulation. There is one important topic called CSF circulation. Later they will discuss it in the gross part. That time you will get to know. Right now remember, the excess accumulation of CSF will lead to a condition called hydrocephalus. Okay. Okay. Can you see the difference here? In the first condition or in the first picture, it is the normal ventricles and foramens filled with the required amount of CSF. But if you observe the second picture, there is an excess accumulation of CSF. As a result, your ventricles and also your foramen is appearing bigger in size. It will lead to the what? This is how the baby suffering from hydrocephalus will appear. Okay. Any doubts in this? Any doubts? No. Okay. Next we will start with the discussion of histogenesis of the neural tube. So, what are all the structures you have seen in the histology of spinal cord? What? Fissures, okay. What about the grey matter and white matter? So, the next topic is histogenesis. Okay. Remember, this is the neural tube. Okay. First, your neural tube will get attached at which somoid level? The fusion of neural tube takes place at which somoid level? At the initial stage, the fusion of your neural tube takes place at the 4th somoid level. Okay. Then the neural tube will first get fold in the cranial aspect, then on the caudal aspect. Once the folding takes place, this is how the structure will look. Okay. So, this part is going to develop as your brain and this part is going to develop as your spinal cord. Okay. So, now we are taking section at this point. Okay. If I take section at that point, this is how the structure will look. Okay, stop talking. So this slit is called as, what it is called as? It is called as dorsoventral cleft. Okay, and this point, this is called as roof plate, and this point is called as floor plate. Okay. On either side of this dorsoventral cleft, on this side we have lateral walls. Okay. This is the gross structure or gross appearance of this cross section. So if I see the same section in a histology aspect, this is how the structure will look. Okay. So this cavity here we called as the dorsoventral cleft, but here we call it as the neurocele. Okay. And this neurocele is lined by which cells? Spinal cord histology is finished, right? Then tell me. Neurocele is lined by which cells? Your neurocele is lined by pseudostratified columnar cells. Okay. So whatever the cells present here, that is your pseudostratified columnar cells, will undergo proliferation and give us two other types of cells. What are those? Your columnar cells will undergo proliferation and give us two different types of cells. What are those? It is neural cells and neuroglial cells. Okay. The neural cells formed here is called as neuroblast cells, and the neuroglial cells formed here is called as spongioblast cells. Spongioblast cells. Okay. Any doubts? So when we take the section and study the histology, we can see the presence of cavity that is called as neurocele, and this cavity is lined by your pseudostratified columnar cells, and this columnar cells will undergo proliferation to give us two different types of cells. Those are called as neuroblast cells and spongioblast cells. So now your neuroblast cells and spongioblast cells will form one more layer around this layer. Okay. So this is the first picture. Around this cell structure we are going to get one more layer. Okay. This is the one more layer. Okay. The first layer is called as matrix cell layer. Stop talking. Okay. The very first layer, that is just your neurocele lined by your pseudostratified columnar cells, that is called as matrix cell layer. The next cell layer, that is formed by your neuroblast and spongioblast cells, that is called as mantle cell layer. Mantle cell layer. Okay. M-A-N-T-L-E, mantle cell layer. Okay. So whatever the neuroblast cells present in this layer will undergo the maturation process. So how the maturation of neuroblast cells takes place? General anatomy. So this is your neuroblast cells. Okay. Once the maturation process starts, it will show the process on both the sides. This is called as bipolar neuron. Bipolar neuron. Okay. So after certain changes, one of the process will disappear and will be left with only one process. This stage is called as unipolar neuron. Okay. Later this unipolar neuron will give rise to your final structure like this. Okay. So this is your axon, these are your dendroids and then the cell body. So now this stage is called as multipolar neuron. Multipolar neuron. This is how the maturation of your neuroblast cells will takes place. Okay. So once the maturation of neuroblast cells takes place, the axons, this part of the neuroblast cells will come out of the mantle layer and forms the one more layer like this. Okay. So this is the neuroblast cells. Once the maturation takes place, their axons will start moving into the next layer like this. Okay. Along with the axons of neuroblasts, some of your spongioblast cells will also move to the next layer. So now the third layer or the outermost layer is called as marginal cell layer. This is how the formation of your 3-cell layer takes place in the histogenesis of the neural tube. Okay. So we have 3 cell layers. The innermost one is matrix and the middle one is mantle, and the last one is, and the outermost one is marginal cell layers. Okay. Your matrix cell layer is made up of neurosil lined by the pseudostratified columnar cells. Mantle cell layer is made up of neuroblast cells and spongioblast cells, whereas the last layer, that is the marginal cell layer, is made up of axons of neuroblasts plus matured spongioblast cells. Okay. Any doubts in this? Any doubts in the neurogenesis? Histogenesis? No doubts? So keeping this cell layer as a basic concept, we have to study the development of your spinal cord and also the development of your hindbrain, midbrain and forebrain parts. Okay. Any doubts? No doubts? Sure? Okay. Then we will start with the development of spinal cord. In which week we can see the development of spinal cord? 4th week or 5th week? 5th? Sure? Okay. So your neurosil, this part, is going to develop as the central canal of your spinal cord. Okay. Or I can say matrix cell layer. Matrix cell layer is going to develop as the central canal of your spinal cord. What is the next layer? Mantle cell layer. So this will give rise to your pons. It will give rise to your grey matter. Okay. And the marginal cell layer, that is the outermost layer, will give rise to your white matter. This is how the formation of your spinal cord takes place. Okay. Your marginal cell layer is also called as ventral zone, okay, and your mantle cell layer is also called as intermediate zone, and the marginal cell layer is also called as marginal zone, okay. We have matrix cell layer which is also called as ventral zone, which is going to develop as the central canal of your spinal cord, okay. The next layer is mantle cell layer, which is also called as intermediate zone, which is going to develop as the grey matter, and the last one is marginal cell layer, which is also referred as marginal zone, which is going to develop as the white matter of your spinal cord, okay. Any doubts? Any doubts in this? So, while studying about the development of spinal cord, we have two important topics to concentrate on. The first one is functional components of the spinal cord, okay, and the second topic is positional changes of the spinal cord, okay. First we will start with the functional components. So, this is how the histology of your spinal cord will look. Not exactly like this, pretty similar to this one, right. So, now this is your central canal part, okay. This is your central canal. So, this is your roof plate. Remember the terms, roof plate, and this is your floor plate, okay. And this end on either side, these are the lateral walls. This one and this one is the lateral walls, okay. This entire structure is divided into dorsal part and ventral part. This area is the dorsal part and this area is the ventral part. This division is made based on the presence of a sulcus in this region. This is called as sulcus limitans, okay. Sulcus limitans, L-I-M-I-T-A-N-S, sulcus limitans. So, sulcus limitans is the structure that divides your entire spinal cord structure into dorsal part and ventral part, okay. The dorsal part is also referred as alar lamina and the ventral part is also referred as basal lamina, okay. Your alar lamina is sensory in function and your basal lamina is motor in function, okay. Next we have a concept called columns. What do you mean by columns? Okay. In your spinal cord, we see only the general columns, okay. Remember this point. In spinal cord, we see only the general columns, whereas in your brain development, we can see both general columns and special columns, okay. So, in this part, if this is a sensory, what type of columns we can see here? Afferent columns or efferent columns? A columns or E columns? Which one we can see? Afferent. A or E? Okay. Afferent columns. So, in the motor region we can see the efferent, okay, efferent columns, fine. So, in your spinal cord totally we can see 4 columns, 2 in alar lamina region and 2 in basal lamina region, okay. I will write it here like this, okay. The very first one is, I already mentioned, we can see only the general column. So, this is general, afferent Stop talking. This is general afferent somatic columns, and the second one is general somatic afferent column and general visceral afferent column. General somatic afferent column and general visceral afferent columns. Okay. The next one, in the basal lamina region, we can see general visceral afferent column followed by general somatic afferent column. So basically the somatic columns are present on the outer aspect, visceral columns are present on the inner aspect. Okay. Remember one more point, your somatic columns are present throughout the spinal cord. Okay. Your somatic columns are present throughout the spinal cord region, whereas your visceral columns can be seen only in the thoracic, lumbar and sacral regions. Okay. Remember this point. So these are the four functional components of your developing spinal cord. Any doubts? Any doubts in the development of spinal cord? First we have seen the development of your central canal, grey matter and white matter from the histogenesis part. Later we have a separate concept called functional components of the spinal cord. In the functional component of the spinal cord, we can see the presence of central canal, basal plate, floor plate, roof plate and lateral walls on both the sides. Okay. And the sulcus limitans is the reference to divide your spinal cord into dorsal part and ventral part. Dorsal part is also called as alar lamina, which is sensory in function. So the columns present here are referred as afferent columns. Okay. And the ventral part is also known as basal lamina, which is motor in function. So the columns present here are referred as efferent columns. Total we can see 4 columns. The first one is general somatic afferent and general visceral afferent, followed by general visceral efferent and general somatic efferent. Somatic columns are present throughout the spinal cord, whereas your visceral columns can be seen only in the thoracic, lumbar and sacral regions. Okay. Any doubts? Any doubts in this? Okay. We can see now notes. Histogenesis of the neural tube. The neural tube increase in thickness due to the repeated mitosis of its epithelial lining. By the middle of the 5th week of embryonic development, the transverse section of recently closed neural tube reveals 3 distinct layers or zones. Those are matrix zone, mantle zone and marginal zones. Okay. So this is the picture for the histogenesis of your neural tube. You can see the formation of all the 3 layers. You can also see the sulcus limitans part. There we have already divided the dorsal area and the ventral area part. Okay. Next, the development of spinal cord. Matrix zone is thick and lies the enclosed cavity called the neurocele. Its numerous cells undergoing mitosis produces the neuroblast cells and spongioblast cells. The former develops into neurons and later develops into the neuroglial cells. The neuroblasts migrate to the adjacent mantle zone. It is the future spinal grey matter, and their axons enter the external marginal zone to form the future white matter. Okay. Once histogenesis is complete, the remaining matrix cells differentiate into ependymal cells lining the central canal of your spinal cord. The development of functional columns. On cross section we can see the cavity which is called as vertical slit. Due to the formation of vertical slit, the dorsal and ventral walls of the neural tube remains thin, that is your roof plate and floor plate. Okay. And are called roof plate and floor plates respectively. The lateral walls of neural tube will get thickened in structure. On each side, the lateral wall of neural tube is demarcated into dorsal and ventral regions by an inner longitudinal sulcus called the sulcus limitans. The cells of dorsal region or alar lamina are functionally afferent or sensory in nature, while those of basal lamina are efferent or motor in nature. Okay? And these are the 4 columns that develops in your spinal cord part. Any doubts in this? Okay. This is a diagram of your cells region, cell layer regions. Okay. Formation of 3 cell layers. Here you can see the formation of sulcus limitans that dividing your dorsal part and ventral part, followed by the adult structure of your spinal cord, similar to your histology of spinal cord part. Okay. You have to mention about all the medians, septums and fissures also here. Any doubts? Any doubts in this? No. We will discuss about one last topic, that is the positional changes of the spinal cord. What do you mean by positional changes of the spinal cord? Okay, I will start with the concept, we can discuss later. So, in the initial stage, that is by the end of 8th week, okay, when your spinal cord is completely formed, this is how the structure will be looking. This is your spinal cord, and to that we have vertebral column. Okay. The cranial end and caudal end of your spinal cord and vertebral column are in same level here during the 8th week. Okay. As the development takes place, by the 24th week, there will be certain changes in the caudal end. Cranial end will remain the same, we can see changes only in the caudal end. Okay. So, now your spinal cord is present at the level of S1 vertebrae. Okay. Next, at the time of the birth, at the time of birth. Okay. This is the spinal cord, vertebral column, spinal cord and vertebral column. Now, your spinal cord is present at the level of L3 vertebrae. Okay. So, at the time of birth, your spinal cord is present at the L3 vertebrae. Then again, we will check for the adult position. Once again, we will check for the adult position or the final position of your spinal cord. So, the final position or the adult position of your spinal cord is, it is present at the level of L1 vertebrae. So, this is the final position of your spinal cord. So, by the end of 8th week, when your spinal cord is completely formed, both your spinal cord and vertebral column are at the same level. After certain development or changes, by the 24th week, your spinal cord is at the level of S1 vertebrae. Then during the time of birth, your spinal cord is at the level of L3 vertebrae. Then after certain development or growth, if we check the position of your spinal cord, it is at the level of L1 vertebrae, and that is the final position of your spinal cord. So, why these changes are taking place? Why the position of your spinal cord is changing during the development process? Okay, anything else? It is because of the changes in the height of the developing embryo. Okay. So, during the 8th week or during the 24th week, your embryo is still in the folding position due to the development of your GIT and also to the development of your brain part. So, during the birth again there will be a single line structure of all the organs present in the body. Later after certain development we will check for the adult position. So, in adults mostly your spinal cord is at the level of L1 vertebrae. Okay. Any doubts? Okay. One last point. So, when your spinal cord is present, your spinal nerves will also present along with the corresponding vertebrae, right. So, what about the adult position? What happens to the spinal nerves present beneath the L1 vertebrae? What are all the nerves we can see? Lumbar nerves, sacral and nerves. These are the three nerves which is present below your L1 vertebrae. Okay. So, whatever the nerves present below your L1 vertebrae will first move in the downward direction and later it will move in the lateral direction like this. Okay. So, the position where your spinal nerves are held, from there is one point where your spinal nerves are held, from there it is going into the downward direction and also towards the lateral point. So, this point is called as, it is called as cauda equina. Okay. So, that position is called as cauda equina. Those structures you can see in the gross part. Okay. Any doubts? So, this finishes your development of spinal cord. You have to write about the histogenesis followed by functional component, positional changes. For clinical anatomy you should mention about the defects of neural tube, that is anencephaly and also spina bifida. Okay. So, this is the columns diagram. Okay. Positional changes of the spinal cord. At the week 8, length of the spinal cord and your vertebral column are equal. The spinal cord extends along entire length of the vertebral canal and the spinal nerve exits intervertebral foramen at the level of its origin. Due to the differential growth of the cord and vertebral column, the intervertebral foramen do not remain at the level of the spinal nerves. In order to exit from corresponding intervertebral foramen, due to the recession of the spinal cord, the spinal nerves are forced to go down in the oblique direction. That means it will first move in the downward direction, then shift towards the lateral side. So, what do you mean by the recession of spinal cord? What do you mean by the word recession? Remains. Okay. Anything else? Anything else? Okay, everyone just go and check once what is recession of spinal cord. It is given both in your embryo textbook and also in your gross textbook. Okay. So, at week 24, the lower end of the spinal cord ends at the S1 vertebrae. During the birth, it will end at the level of L3 vertebrae. So, the adult position is the lower border of the L1 vertebrae. The nerve roots that descends below the lower end of spinal cord, surrounded by the thin thread-like prolongation of pia mater, which forms at the tip of the conus medullaris to form the cauda equina. So, you have to remember about the cauda equina and all the 4 positions. The first one is at the same level, second one S1, third one is L3 and the final position is L1 vertebrae. This is all about the development of your spinal cord. Okay. Any doubts? Okay, we have discussed about the ventricular system and its foramens, followed by the clinical conditions. We have discussed about 3 clinical conditions, then the histogenesis of the neural tube, that is formation of 3 cell layers, followed by the development of your spinal cord. Okay.